I'm Kalpana Madhushani from University of California, and today my presentation topic is spatial distribution of water quality parameters in the Malala Lagoon in Bundala National Park, Sri Lanka. First, let's move on to a brief introduction. The Bundala National Park is the first Ramsar wetland in Sri Lanka. It is very important due to significant wildlife diversity and most importantly, it is a wintering ground for migratory birds. It is also considered as a man and biosphere reserve. Malala Lagoon is situated in the south coast of Sri Lanka and it is the largest lagoon of Bundala National Park. It also supports the migratory and residential fauna. However, the physiochemical characteristics of this lagoon have largely been influenced recently due to the upstream irrigation work, especially the Lunugahambahara irrigation scheme, where the drainage water partly released into the Malala Lagoon. So, the presence of invasive plants in this lagoon is commonly attributed to the ecological changes that possibly have resulted from altered water salinity caused by the increased freshwater input into this lagoon. The three images bottom of the slide shows the extensive distribution of type angustifolia in the Malala Lagoon. So, objective of this study was to investigate the current status and the spatial variation of water quality in Malala Lagoon. So let's move on to the methodology. Here we collected water samples randomly from 15 locations throughout 3 strata of the lagoon as Malala North, Malala Middle and Malala South. And the samples were conducted twice in both dry season and wet season during 2018 and 19. The sampling locations were recorded with Garmin GPS name. The pH and the electrical conductivity was measured using a portable meter, and then the samples were treated at the field and transported to the laboratory for analysis of dissolved nutrients. Nitrate nitrogen, ammonia nitrogen, and total nitrogen, orthophosphates and total phosphates were analyzed using preserved samples according to the AFA 1998 methods. And finally, these parameters were interpolated using RMAP 10.2.2 software and SPSS 22 software to determine the spatial distribution. So let's move on to the results. Here you can see the overall water quality in Malala Lagoon during the study period. Here the nitrate nitrogen concentration was recorded as 0.4313 milligrams per liter and the ammonia nitrogen concentration was recorded as 0.3072 milligrams per liter and the total nitrogen concentration was recorded as 1.0567 milligrams per liter. Orthophosphates were recorded as 0.0147 milligrams per liter and the total phosphorus was recorded as 0.0306 milligrams per liter. And the electrical conductivity was recorded as 1283.02 microsiemens per centimeters and the pH was recorded as 7.78. This image shows the spatial distribution of different nitrogen forms in the Malala Lagoon during the study period. Here you can see that the higher concentration of all the nitrogen forms, like nitrate nitrogen, ammonia nitrogen, and total nitrogen, higher concentrations were recorded in the northern section of the lagoon. And this is the spatial variation of different phosphorus forms in the Malala Lagoon. And here also you can see that higher concentration of orthophosphates and total phosphates were recorded in the northern section of the lagoon. This is the seasonal variation of water quality in the Malala Lagoon. And here you can see higher concentration of all the nitrogen and phosphorus forms were recorded in the northern section, and also higher concentrations were recorded during the wet season than dry season. This is the seasonal variation of electrical conductivity and pH. Here you can see in both seasons lower concentration of electrical conductivity and pH were recorded in the northern section. However, the higher concentration was recorded in wet season than the dry season. So according to the results, we can conclude that the highest concentration of all the forms of nitrogen and phosphorus were observed from the northern part of the Malala Lagoon because this is the area that receives the drainage irrigation water. So this higher concentration gradually decreases towards the southern part of the lagoon, which is closer to the outlet. The lower electrical conductivity and salinity in the northern section of the lagoon is also due to the dilution of concentration with the excessive fresh water that enters the lagoon. And finally, the present study provides an insight into the spatial distribution of water quality parameters that manifest the anthropogenic impact on the lagoon ecology. So these are my references. And we acknowledge National Science Foundation of Sri Lanka for funding this project. Thank you.